Thank you kindly, Annika, for that introduction and for the invitation to come back here to St. Anne's. As Annika mentioned, I had the pleasure and privilege of serving as a junior research fellow for 4 years, from 2018 to 2021 here at the center. And during that time, I got to support various CPM initiatives ranging from high-profile lectures, such as this Dr. Stanley Ho Memorial Lecture, which featured the likes of Dr. Jennifer Doudna, who received the Nobel Prize for her work in discovering the CRISPR-Cas9 genome editing system. I helped organize research symposia addressing issues such as metabolic health and facing up to disparities in healthcare. And then I really enjoyed supporting student-led initiatives such as the then burgeoning Oxford University Personalized Medicine Society, which was spearheaded by students here at St. Anne's, as well as the vitally important work of extracting DNA from strawberries, which was good fun. I enjoyed it. The primary students in the room seem to enjoy it too, but not as much as my fellow ex-JRF, Dr. Porrick Dixon, shown on the bottom right and seated right there. As many of you know, the Oxford Center for Personalized Medicine is a vehicle to promote discussion of scientific advances in the field of personalized medicine, as well as dealing with some of the challenges that come with implementing these advances into the clinic. And so it seems like a sensible place to start would be defining personalized medicine. What is that? Well, if you Google or ChatGPT, a typical definition you might find is that personalized medicine is the area of medicine that aims to tailor healthcare practices to individual patients based on their genetic, environmental, and lifestyle factors. Now, in face value, this is pretty much the aim of medicine in general. What really sets personalized medicine apart is its focus on leveraging recent advances in genomics, biotechnology, molecular phenotyping, data analytics to improve the process of medicine, which is diagnosing disease, treating disease, predicting disease onset, and ideally preventing disease altogether. It recognizes that people vary considerably in their vulnerability to disease, as well as how they respond to treatment. And an important source of this variability is genetics. And in fact, much of the foundation of what we consider now to be personalized medicine comes from the field of genomic medicine, with sub-areas like pharmacogenetics, where we consider genetic variation that influences how patients respond to medicines. A well-known example would be variants in the VCORC1 gene, which influence patient sensitivity to the anticoagulant drug warfarin, as well as variation within the gene CYP2C9, which influences how that drug is metabolized in the liver, both of which factor into dosing strategies. But genetics is also important, can be very important when it comes to diagnosis and treatment. The classic example here would be the disease cystic fibrosis, which is a genetic disorder resulting from single gene mutations in the gene CFTR, which encodes a chloride ion channel present in the respiratory system and the pancreas. Hallmark symptoms would be buildup of mucus in the airways with impaired lung function and higher risk for infections. Now, the specific variants involved can help inform diagnosis of the particular subtype of cystic fibrosis, as well as potential therapies. For example, the glycine 551 aspartic acid substitution results in a gating mutation in the channel of this chloride ion transporter. And such mutations or cystic fibrosis cases resulting from these type of mutations tend to respond better to a class of drugs known as potentiators, such as ivacaftor, that restore that normal gating function. On the other hand, this phenylalanine 508 deletion actually impairs proper folding of this protein, which impairs its ability to position correctly within the cell membrane. And cystic fibrosis resulting from this type of mutation responds much better to a class of drugs known as correctors, which allow that protein to fold properly, make its way to the cell surface, and carry out its normal function. So genetics can certainly be important when considering disease, but when considering any particular disease, it's worth considering what's known as the genetic basis of that disease. So what does that mean? Well, we might ask questions like, to what extent is this disease inheritable? That means, is this disease influenced by genes that are passed down from parents to children? Well, this is certainly the case for cystic fibrosis. That's an autosomal recessive disease. It requires inheriting 2 copies of risk variants from each parent. This is in contrast to, let's say, a cancer that results from exposure to carcinogens, mutagens that damage DNA, cause disruption in oncogenes and tumor suppressor genes, resulting in unregulated cell growth and the development of tumors. This is certainly genetic, but it involves mutations that are neither inherited from parents nor passed down to children. Another term that often gets confused with whether or not a disease is inheritable is this term heritability, right? Heritability is a population parameter that describes the extent to which observed phenotypic differences in a population are due to genetic differences. And a classic example here is height, where in a population, up to 70% of the observed differences in individuals' heights are due to genetic differences. But there are diseases with high levels of heritability. Type 2 diabetes, coronary artery disease, both have heritabilities reported to be in the range of 50%. But a related concept that I would like to especially focus on today is a concept known as the genetic architecture of disease. You encounter this a lot now. And what is meant by genetic architecture is characteristics of the genetic variation responsible for these traits, right? So these characteristics include the number of risk variants. Are we dealing with a single gene disorder like cystic fibrosis? Or is it a disease like type 2 diabetes, where you have over 1,000 variants that each contribute to disease risk? Such a disease is called polygenic as opposed to monogenic. We think about the type of variants involved. Is the disease mostly due to single nucleotide substitutions, or are there structural variants that involve insertion or deletion of large DNA sequences? We think of the magnitude effects. In the case of cystic fibrosis, phenylalanine 508 deletion has a very strong effect, whereas any particular variant that increases your risk for coronary artery disease is pretty mild or modest. Frequency, are these common variants? Are they rare variants in a population? And importantly, interactions, right? If we think about a particular risk variant, is its risk effect modified by the presence of some other variant in the genome? Or is there an environmental exposure that might increase or decrease the risk of that variant? All of these come into play when we think about genetic architecture. Now, one plot that gives you some insight into genetic architecture is what's known as a Manhattan plot. And these are often used or shown in the context of genome-wide association studies, or GWAS. This has been the predominant study design in human genetics for the past 20 years now. And basically it involves, assembling large groups of unrelated cases and control individuals, genotyping them at hundreds of thousands to millions of markers, and then testing for association. You look at results across the genome, and wherever you find a locus that strongly associates with disease risk, you have a strong peak, right? That resembles a skyscraper in a city skyline. It's Manhattan plot. And here, in this case, we're looking at a typical Manhattan plot for kidney stone disease, and there are 20 loci that associate with disease risk. Gives you some insight into the architecture of this trait. But what I really want to focus today on is that most of what we know about the genetic architecture of complex traits and diseases has been discovered in populations of European ancestry. European descent. Non-European populations are starkly underrepresented in contemporary genetics research. There's this nice site called the GWAS Diversity Monitor, which keeps an up-to-date record of participants involved in these GWAS studies. And as of 2 days ago, 93% of participants in GWAS studies are of European descent. And this is a bit out of step with global proportions. Right, European descent individuals make up less than 20% of the global population, but well over 90% of participants in GWAS. In contrast, Latinos, Hispanics, Latin Americans make up of almost 10% of the global population, but comprise less than 1% of GWAS participants. So this is an issue, and it's been a long-standing problem in human genetics. You can see here on the right that this is nothing new. But this is not a problem for the sake of representation. This lack of diversity actually undermines genomic medicine. And it's worth pointing out that it is possible for risk variants to be present in a non-European population, or even specific to that non-European population. And this variation in causal mutations can actually confound diagnosis or treatment. If we go back to cystic fibrosis, I mentioned that the phenylalanine 508 deletion. This mutation is responsible for up to 70% of cystic fibrosis cases in individuals of European descent, but only 29% of cases in those of African descent. Moreover, there's this kind of a complicated name for a variant, this 3120 plus 1G to A substitution, intronic variant. This is responsible for up to 65% of cases in South Africans of African descent. And in fact, not considering such variants that might be segregating at higher frequencies in non-Europeans could result in underdiagnosis of these diseases when applied outside of Europe. Another point that I want to introduce is that when trying to diagnose rare genetic disorders, it's important to identify, ideally, the causal variant responsible for these diseases, a given disease. And one criterion used to determine whether or not a variant is causal is whether or not it's present among healthy individuals. So you can have a variant that is completely benign, right? No impact on disease, that is common in non-Europeans, but rare in Europeans. And if your reference data set is all Europeans, then you might think that this perfectly benign variant is actually causal if you don't have the full spectrum of allelic variation present in our reference sample. So this could result in mistaken inferences about what the causal variants are. And again, a lot of the genetic prediction tools we've developed have been based on European studies informed by European genetic architectures. And this could impair transferability of genetic prediction across non-European populations, which can actually worsen health disparities that currently exist in healthcare systems. So it's therefore important to do studies outside of Europe and European descent populations. With that said, I'd like to introduce the cohort I work with, the Mexico City Prospective Study, or MCPS. This is the largest blood-based prospective study in Latin America. It was started over 20 years ago by epidemiologists working at the National Autonomous University in Mexico City, and they forged a collaboration with epidemiologists working here at the University of Oxford, and were initially interested in looking at the impact of smoking on public health in Mexico. At the time, it was known that smoking is a leading cause of death, but most of those studies were done in high-income countries. But this plan evolved to move beyond smoking and eventually design a prospective study to explore not only smoking, but other exposures that could lead to disease and premature death in Mexico. Between 1998 and 2004, over 150,000 adults were recruited from two districts in Mexico City, the centrally located district of Coyoacán and the adjacent and densely populated district of Iztapalapa. After obtaining informed consent from each participant, field workers administered a questionnaire that included items pertaining to previous diagnosis of disease, medication use, socio-demographic variables, and lifestyle factors. They took physical measurements, which included blood pressure, height, weight, BMI, took a 10 milliliter blood sample from each participant, and importantly, have linked these samples to a national death registry, which is centrally reviewed and adjudicated and has information on primary and contributing causes of death. Now, if this study was done today, researchers would use available technology now, like an application on a smartphone or iPad to administer the questionnaire. Back then, they used a barcode reader, which was actually pretty reliable and accurate. Here's an example of what a question on the questionnaire would look like pertaining to alcohol consumption, number 45. In the last 12 months, how often did you drink any beverage with alcohol, such as beer, pulque, wine, tequila? So the participants would have seen a question like this, and the field workers would this would have been the set of options available to them, and they would scan the corresponding barcode for the answer. Now, data collection ended in 2004, but in a prospective study where you sample from a general population without any particular ascertainment for a disease or a condition, an important ingredient is time, right? Once you collect samples, you just wait for life to go on, and over time people eventually get sick and some die, and as a researcher you wait to some point in the future to then go back, observe the number of deaths that have occurred, and try to work out what were the major risk factors responsible for premature death over that time period. And so for 12 years nothing much happened in the study, but starting at around 2015, MCPS investigators really started to get some insight into the major causes of disease and premature death in Mexico. There have been numerous studies that have been published in recent years. For example, going back to smoking, maybe no surprise to you that smoking is an important cause of death in Mexico. However, it's not quite the same as what's been observed in other populations. Now, smoking rates are high in Mexico. About 1/2 of men and 25% of women reported smoking at baseline. But smoking patterns, cigarette consumption is low in Mexico, and it's also low in other low and middle income countries. In high income countries, it's not uncommon, at least at that time period, for smokers to consume an average of about 15 to 20 cigarettes per day. But in Mexico, 1/3 of smokers reported smoking cigarettes on a non-daily basis. So there's a higher prevalence of low intensity smoking, but still smoking even a few cigarettes per day increases mortality among smokers that smoke less than 10 cigarettes a day, of which averaging about 5. There is a 17% increase in all-cause mortality within the 12-year follow-up period. So smoking is an important cause of death in Mexico, but it's not the most important cause of death in Mexico. That is diabetes. At baseline, by age 60, 20% of all participants had a previous diagnosis of diabetes. For context, that's more than double the prevalence of diabetes reported in the US and the UK at that time period. When we look at mortality, if we just focus on, let's say, adults aged 35 to 59, individuals that had a previous diagnosis of diabetes at recruitment were more than 5 times likely to die of any cause within 12 years. Across all adults from 35 to 84, the risk ratio was over 4, which is more than double the death rate attributable to diabetes observed in the UK and the US at that time period. So clearly, MCPS has provided valuable insights into causes of disease and premature death in Mexico. But the MCPS investigators have also maintained a longstanding interest in looking for genetic risk factors for disease in this cohort. It's just that 20 years ago, we were still at the tail end of the Human Genome Project, and we're many years away before having the technology to genotype a cohort of this magnitude, let alone in a cost-effective way. That didn't happen until 2020, when the investigators formed a partnership with the pharma companies AstraZeneca and Regeneron to make available genome-wide genotyping data. And currently, we have genotyping array data available using the Illumina GSA Global Screening Array version 2 for over 140,000 participants in the cohort. Exome sequencing, which is a sequencing method that enriches for the 2% of the genome that actually encodes proteins, protein encoding genes. And this is available for 141,000 individuals. And importantly, we have whole genome sequences for a subset of 10,000 individuals, immediately making this one of the largest genetic studies in a non-European population. I joined in 2020, which is not a bad time to start a job in this project. And in the following years, I worked closely with colleagues in Mexico and at the Regeneron Genetics Center to carry out a comprehensive genetic profile of MCPS. And so, as you can imagine, there are many different analyses you could explore with this data. One of the first things you can do with genetic data is look at genetic relationships present in the cohort, and of which there are many. So we found that in this cohort of 140K individuals, there were over 31,000 parent-offspring pairs embedded in the study, over 29,000 full sibling pairs, nearly 3,600 nuclear families, and overall, we estimate that 71% of participants had at least one close relative in the cohort within the 3rd degree. So a lot of relatedness. We can come back to why that is, but that was much more than we expected. The largest 1st-degree family network is shown here, where there are 48 MCPS participants that share a 1st-degree relative. And when we build 2nd-degree family networks, it can actually be quite extensive. So if this plot, each point is a person, right? And each line is a relationship between them. And what was surprising to us is the largest embedded 2nd-degree network in this cohort has over 9,000 individuals. So there's extensive relatedness in the study. And as you can imagine, from a genetics perspective, there's quite a lot you can do with genetic relationships. But another phenomenon we were interested in characterizing is what's known as population structure. It has a technical definition. It's systematic differences in allele frequencies among subpopulations within your cohort. And to maybe make that a bit more meaningful, we have these plots here. This is results from a high dimensional data decomposition method known as principal components analysis, decomposing genetic data, where each point is an individual in the MCPS cohort. And where they cluster, where they position on this plot is by genetic similarity with each other. And using this data, in addition to reference data from populations around the world, we identified a subset of individuals that have high levels of genetic similarity with Southern Europeans. But we also find subsets of individuals with high levels of genetic similarity with contemporary indigenous communities throughout Mexico. This cluster here in purple shows a high level of genetic similarity with indigenous communities from the southeastern part of the country, including the Yucatan Peninsula and the state of Chiapas. This is the historic site of the Mayan Empire. There is, of course, high levels of genetic similarity in this cluster with individuals from central Mexico, surrounding Mexico City. This again is the historic site of the Aztec Empire, as well as communities in the Lake Texcoco region surrounding Mexico City. And interestingly, we found two subclusters of individuals with genetic similarity to Southern indigenous communities from the state of Oaxaca on the south coast. So you can see that there is extensive population structure, but also a phenomenon known as admixture, where the genome of a typical person in MCPS can be understood as a mosaic of genetic sequences inherited from both European and indigenous American ancestral populations. So we've done an analysis known as local ancestry inference to work out these mosaics. And on the left, we have a karyogram for an individual that is primarily of African descent with some indigenous American admixture. This person in the middle is highly admixed with contributions from both indigenous American and European ancestry. Overall, when we look at autosomes in the data, we see that there's about 66% average indigenous American ancestry in this cohort. But there are deviations from that. If we look at chromosome X, the average indigenous contribution is 79%. This is something that's been reported elsewhere, and it's a phenomenon known as sex-biased gene flow, which is a direct consequence of European colonization of the Americas. So we've characterized these features in the cohort, but importantly, with the exome and whole genome sequencing data, we performed a comprehensive survey of variation. So within the exome sequencing data available for 141,000 individuals, we found 4 million coding variants. So these are variants within the 19,000 genes present in the genome. Most of these were rare, meaning they had a minor allele frequency less than 1%. But notably, more than a fourth of these variants were unique to this cohort, had not previously been reported in large-scale European studies like the UK Biobank or the NOMAD study. And of these variants, I want to just point out here, there were about 230,000, is that right? 230,000 predicted loss of function variants or PLOF variants. These are genetic variants that are predicted to have severe consequences on proteins, severely altering function or truncating the protein so that they lose their structural integrity. For example, creating a premature stop codon that results in a truncated protein. So that's quite sizable. When we look at the whole genome sequencing data, there were over 131 million variants, about a tenth of which, 31.5 million, were also unique to MCPS. So again, this enhances diversity in terms of the variants present. And as mentioned earlier, having such variants available in our references can be useful for diagnosing rare diseases for anyone, regardless of their ancestral background. And so to facilitate this type of use, we worked with our colleagues at Regeneron to create this MCPS variant browser that's freely accessible now. You can, for any given variant, look at its allele frequency in MCPS, as well as its inferred ancestry-specific allele frequency in African, European, and indigenous American backgrounds. This work was published recently in Nature. It was nicely featured on the cover, alongside another study led by a completely independent group in Mexico called the Mexico Biobank. I think that's another really cool study. And collectively, both of these studies provide valuable insight into genetic variation present in Mexico, which has largely, for a long time been underrepresented in genetics research. But how has this helped us understand the genetic basis of disease? So for any given disease, it's useful to know what genes are actually involved in mediating disease risk. And one line of investigation that's proved fruitful in the past few years has come from studying rare genetic variants. More and more researchers are interested in studying exome data to conduct gene-based tests, focusing on what are known as qualifying variants. These are rare deleterious variants, just like the PLOFs I mentioned earlier, right? Variants that cause severe damage to the structural integrity or alter the function of proteins. What you do is develop an association test where for a given gene, you identify the number of protein truncating variants in cases and controls and test for association with diseases. The advantage with these type of tests is that they directly implicate genes involved in the biology of the trait, and they can potentially identify drug targets, as we'll see. So in recent years, we've contributed to a number of studies looking for rare variant burden with various outcomes. So we participated in the largest exome study of obesity recently, involving over 640,000 participants, testing for rare variant burden at the gene level, and found that there were 16 genes that associated with body mass index. Of them, notably, was a gene that hadn't been previously reported, known as GPR75. This is a G-coupled protein receptor that's highly expressed in the hypothalamus of brain tissue. And what we found is that carriers for this carriers for protein truncating variants at this gene had lower levels of BMI on average. Now, these are rare variants present in only 4 out of every 10,000 people, but those that had protein truncating variants in this gene were found to have lower levels of BMI. Such variants associated with 1.8 kilograms per meter squared lower BMI and a 54% lower odds of obesity. Our colleagues at Regeneron performed a series of perturbation studies for the homolog gene in mice, where they perturb either 1 or 2 copies of this gene, subjected mice to a 2-week high-fat diet challenge, and found that when impairing this gene, mice with the perturbation were observed to have, they didn't report much, 25 to 44% reduction in weight gain relative to wild-type mice. And this also corresponded with a better insulin tolerance and sensitivity profile. So as a drug target, this is interesting. It's easier to turn off a drug than turn on a drug, or sorry, turn off a gene than turn on a gene, which makes this a potentially interesting drug target for obesity. We followed up the study with a separate effort looking at fat distribution rather than overall BMI. In this case, we found that there were 16 genes where protein truncating variants associated with differences in waist to hip ratio adjusted for BMI, including another novel gene, inhibin beta E, where protein truncating variants in this gene associate not only with lower fat distribution, but also lower odds of developing type 2 diabetes. I mentioned smoking earlier. Last year, we contributed to the largest rare variant study of smoking, and found that rare protein truncating variants in the gene CHRNB2 significantly reduced the likelihood of smoking. Notably, this gene encodes a nicotine acetylcholine receptor expressed in the brain that's known to facilitate the neuronal responses to nicotine. But as I mentioned, the leading cause of death in Mexico is not smoking, it's diabetes. And we were very interested to contribute to a study looking for rare variant burden for diabetes outcomes. This was a study led by colleagues at AstraZeneca, which looked at rare variant burden in the UK biobank. For those not familiar, this is a large cohort of nearly 500,000 individuals from the United Kingdom, predominantly white British descent. And in this study, they found a number of genes that associated with diabetes risk. Many of these genes have been previously reported in other studies or implicated in monogenic forms of diabetes, like glucokinase and HNF1 alpha. But there was this one gene, MAP3K15, which was found to have an association with lower risk for diabetes. And when we look at MCPS for replication, we similarly found that protein truncating variants, again, variants that disrupt the integrity of this gene, conferred protection against diabetes. In this case, both type 1 and type 2 diabetes. And again, you might think this could be a potentially novel drug target, but it's also worth considering other genetic factors that might be at play. And in the case of Mexico, a study 10 years ago, led by the Sigma Consortium, studying about 7,000 sequences in Mexico, found that risk variants in this gene SLC16A11 are very common in Mexico and had higher effects than you would expect based on evolutionary theory. What I'm showing here are what are known as haplotypes. So instead of looking at a single variant, you look at a set of variants that are inherited together. And on the bottom, we have this risk haplotype, where 5 risk alleles at 5 different variants are inherited together. Now, this risk haplotype is rare in Africa and low frequency in Europe, but among Mexican, Americans, and Mexicans, this haplotype is present in about 30% of individuals, and among Mexicans with more than 95% Native American ancestry, it's present in almost half of individuals. Now, I've recently run a GWAS and MCPS for type 2 diabetes, and as we can see, variants at SLC16A11 also replicate here, strongly associate one of our strongest associations with diabetes. So when we come back and look at MAP3K15, right, this gene where if we disrupt it, it confers a protective effect. If we look at it in relation to SLC16A11, which is common in Mexico, things are a bit more complicated. So if we just look at the bottom here, these are individuals that don't have that SLC16A11 risk haplotype, but do have protein truncating variants in MAP3K15. And among these individuals, you see a protective effect of protein truncation in MAP3K15, similar to what you find in white British individuals from the UK biobank. But among individuals that have the SLC16A11 risk haplotype, which is present in 30% of individuals in Mexico, that protective effect is offset. So this, as you can imagine, could have potential clinical implications if you're targeting MAP3K15 when considering the genetic background of a given individual, which may involve variants that are segregating at different frequencies across populations. So I alluded earlier to the idea of leveraging genetics to predict disease risk. And this is something that's been tremendously popular over the past 7 years or so. If you've taken any of these direct-to-consumer genetic tests from companies like Ancestry.com or 23andMe, you'll no doubt have risk profiles regarding your risk of developing heart attacks or diabetes or what have you. These are based on what are known as polygenic scores or polygenic risk scores. There's actually a really nice explainer on the CPM website, animation if you want to check that out. But basically, it involves combining patient genotypes with results from studies like GWASs, kind of reference data. What you do is you combine this information to calculate a single score for each individual that quantifies their polygenic burden for complex traits like type 2 diabetes, coronary artery disease, and dementia. And the reason why they've become so popular is there have been a number of studies that show they can actually be quite helpful in predicting at least some outcomes. There was a seminal study led by Sakar Katharesan's group at the Broad in 2018 that found that there was a subset of individuals whose polygenic burden conferred a risk of over threefold. In other words, 8% of individuals had this polygenic burden that increased their risk of coronary artery disease by over threefold. And again, this is based on common variants, most of which have small effects individually on disease risk. But when combined, the overall genetic risk was comparable to known rare monogenic mutations that are known to considerably increase disease risk. So this raised a lot of eyebrows and has since been replicated and corroborated, not just for coronary artery disease, but for other complex diseases as well. And it's worth emphasizing that they're already making their way into the clinic. So there are health systems in the United States, the Mayo Clinic, the Geisinger Health System, the North Shore Health System in Chicago, that are using polygenic risk scores in the clinic. And currently, there's this ongoing study here in the United Kingdom called the Our Future Health Study, which is aiming to recruit 5 million participants from the United Kingdom. And part of the study will involve genotyping all participants, calculating polygenic scores for diabetes, coronary artery disease, dementia, cancers, stroke, and using this to generate an individualized risk profile that will be used to guide treatment, prevention strategies, and potential genetic screening. So this is already making its way into the clinic. But it's been known for quite some time now that polygenic scores yield inconsistent predictions across populations. Right? This is a study that Alicia Martin published in 2017, but it still holds today. If you were to calculate a polygenic score for height using European-based data, you find that it predicts lower genetic height in non-Europeans. Similarly, a genetic risk score for schizophrenia based on European studies particularly underestimates polygenic burden for African descent individuals, despite similar disease prevalences between Europeans and Africans. And then when we look at type 2 diabetes, the relative order of the polygenic burden distribution varies depending on the source of the genetic data used. So you could imagine that using genetic information that is systemically biased against particular populations has issues when you think about clinical utility, and it risks exacerbating health disparities. This is something we've explored a bit in Mexico. And for example, here, when we calculate a polygenic score for body mass index using data from the UK Biobank, which again is predominantly of white British representation, we see that as Indigenous American ancestry increases, the ability to predict height in this cohort decreases. We've done a reciprocal analysis where we instead generate a polygenic score using information from the Mexico study to predict BMI among different ethnic groups in the UK Biobank. And you can see that the highest prediction corresponds to the subset of Latino individuals in the UK Biobank. Now, the confidence interval is massive, right? And this is reflecting the fact that there are less than 1,000 Latinos in a study of 500,000 Brits, right? So this definitely underscores the fact that there is a need to, again, increase representation of non-Europeans in genetic studies. So with that said, I will conclude by, again, reiterating that much of what we know about the genetic architecture of complex traits and diseases has been inferred from studying European descent populations, and this lack of representation undermines personalized medicine in a number of ways. We miss population-specific risk factors like SLC16A11. This impedes diagnosis of rare genetic disorders because we don't have the full spectrum of genetic variation in our references. It impairs genetic prediction, as we've just seen, and taken together, this could all worsen health disparities that already exist in the US and the UK. Therefore, including globally diverse populations such as MCPS are essential to addressing this problem, but myriad challenges remain, both in terms of scientific and analytic issues with dealing with results from different genetic architectures to improve polygenic prediction or identifying causal variants. There are logistical challenges for conducting studies in low and middle income countries, which our colleagues in Mexico know very well. And then, of course, there's social and ethical issues with ensuring that research done in low and middle income countries are done in ethical ways and ensure equitable benefits. So with that said, many people to thank contributing institutions and funders, and I would like to thank the MCPS investigators. Shown here on the far right is Professor Jesus Alegre Diaz, who's been part of the study for over 20 years. He's shown here with his field workers. They're currently gearing up to recontact individuals to enhance the study. And it's just worth pointing out that, between 1998 and 2004, they knocked on 100,000 doors in Coyoacán and Iztapalapa. I mean, that's not a trivial thing. So we're grateful to him and his team for leading that effort. And of course, the 150,000 people that answered those door knocks and agreed to be part of the study. So with that said, thank you.